Welcome, everyone. I'm excited to kick off our presentation today. Today, we'll be exploring Biomolecules. This is the first slide, so just a quick welcome and overview. We'll look into what biomolecules are, their types, and why they are essential for life. So, let's get started and dive into the fascinating world of biomolecules. Today, we are going to explore the structure of nucleic acids, which are essential biomolecules carrying genetic information. We will also look at protein denaturation, a process where proteins lose their functional shape due to environmental changes. Interestingly, some molecules, like certain sugars, lack an asymmetric carbon, which influences their structure and classification. We'll briefly summarize key concepts, including starch, which is a storage form of glucose in plants, and the structure of fructose, a simple sugar with the chemical formula C6H12O6. We'll discuss D and L notations, which are based on the structure of glyceraldehyde, helping us understand stereochemistry in sugars. Next, we'll classify carbohydrates based on their nature, differentiating between monosaccharides, disaccharides, and polysaccharides. This leads us to vitamins, vital nutrients that support various bodily functions. We'll compare fibrous and globular proteins, highlighting their structural differences and functions. The classification of amino acids will also be covered, emphasizing their role as the building blocks of proteins. Then we'll delve into polysaccharides like starch, glycogen, and cellulose, exploring their structures and biological roles. We'll touch on concepts like anomeric carbon and anomers, which are crucial in understanding sugar chemistry. The mechanism of enzyme action will be explained, illustrating how biological catalysts accelerate reactions. We'll summarize the key points in a nutshell, tying everything back to the importance of biomolecules. Next, we consider nitrogenous bases, essential components of nucleic acids, and the classification of proteins based on their structure and function. We'll cover proteins and enzymes, focusing on their significance in biological processes. Moving on, we'll look at glycogen, the animal storage carbohydrate, and the structure of proteins, which are vital for cellular function. The concept of mutarotation will be introduced, explaining how sugars exist in different forms. The importance of carbohydrates in nutrition and energy storage will be emphasized, along with the preparation and structure of glucose, our primary energy source. We'll also examine cellulose, an important structural component in plants. Finally, we'll revisit the classification of carbohydrates, the cyclic structure of glucose, and the classification of monosaccharides, tying everything together with a modern definition of carbohydrates. We'll also include a quick review to test our previous knowledge, ensuring a comprehensive understanding of these fundamental biomolecules. Now, let's explore why we prefer a balanced diet. A balanced diet provides all the essential nutrients our body needs in the right amounts. This helps us maintain good health, stay energetic, and prevent diseases. When our diet includes carbohydrates for energy, proteins for repair and growth, vitamins for various body functions, and minerals, it supports overall well-being. Understanding the composition of the human body is also crucial. Our body is made up of water, proteins, fats, minerals, and carbohydrates, with DNA and RNA playing vital roles in genetic information transfer. Speaking of genetics, transference of genetic information from one generation to another occurs through DNA, which carries our hereditary traits. And finally, to answer how we transfer genetic information, it happens during reproduction, where DNA from parents combines to form new genetic combinations in their offspring. This process ensures continuity of traits across generations. All these points highlight the importance of a balanced diet in supporting our body's complex functions and maintaining our health. A living system, as you know, is something that grows, sustains itself, and can even reproduce. It's quite fascinating because, at its core, it's made up of non-living atoms and molecules. This might seem counterintuitive. How do non-living parts create something alive? Well, that's where biochemistry comes into play. It's the science that helps us understand the chemical processes happening inside living organisms. Living systems are composed of various complex biomolecules, such as carbohydrates, proteins, nucleic acids, and lipids. These molecules are fundamental to life. For example, proteins and carbohydrates are not only essential components of our diet, but also perform crucial roles within our bodies. They interact in intricate ways, forming what we might call the molecular logic of life processes, kind of like a complex code that keeps everything functioning smoothly. Additionally, simple molecules like vitamins and mineral salts might seem small or insignificant, but they are vital for proper biological functions. They support everything from metabolic reactions to immune responses. All of these elements, these biomolecules and small molecules, are interconnected, creating the dynamic and complex system that is life. It's a perfect example of how something seemingly simple at the atomic level can give rise to the extraordinary complexity of living organisms. Let's take a look at this sequence that links biomolecules to living organisms. It all starts with biomolecules, which are the basic organic compounds that are vital for life. These include things like carbohydrates, proteins, vitamins, and lipids. Even though they are considered lifeless, they form the foundation of all living systems. They are responsible for growth, repair, and maintaining the body's functions. From these biomolecules, we move on to cells, which are the smallest units of life. Cells build tissues, which are groups of similar cells working together. These tissues form organs, like the heart or lungs, each with specific functions. Finally, all of these components come together to make up a living organism, a complete, functioning living being. So this sequence illustrates how tiny, lifeless molecules are the starting point for the incredible complexity of life we see around us. Now, it's important to realize that not every compound fitting into the general formula we discussed earlier can actually be classified as a carbohydrate. For example, acetic acid, which has the chemical formula CH3COOH, fits into the formula C2H2O2, but it's not a carbohydrate. Similarly, rhamnose, with the formula C6H12O5, is indeed a carbohydrate, but it doesn't quite fit that same general formula. Most compounds we consider as carbohydrates share a common feature. They generally follow the formula CX, H2O, which is why they were historically called hydrates of carbon, or hydrates of carbon, based on that old definition. For instance, the molecular formula of glucose, C6H12O6, perfectly fits into this pattern as C6H2O6. Examples of carbohydrates include common substances like cane sugar, glucose itself, and starch. These compounds are primarily produced by plants and make up a huge group of naturally occurring organic molecules. So, to sum up, while many compounds follow this general formula, only certain ones truly qualify as carbohydrates based on their structure and origin. And understanding this distinction helps us appreciate the diversity and significance of carbohydrates in nature. Let's take a closer look at what carbohydrates are. Essentially, they are compounds that include optically active polyhydroxyaldehydes or ketones. These are molecules that have multiple hydroxyl groups attached, and they can be either aldehydes or ketones. Importantly, some compounds produce these units when they undergo hydrolysis, meaning they can break down into simpler carbohydrate units. Carbohydrates are also commonly known as saccharides, which you might have heard before. Now, when we talk about the modern definition of carbohydrates, we're emphasizing their chemical structure and behavior. They're not just simple sugars, but can be complex molecules that contain multiple hydroxyl groups and carbonyl groups, giving them specific optical activity. Understanding this helps us see how carbohydrates are integral to many biological processes, from energy storage to cell signaling. They're fundamental molecules in our bodies and in nature, tying into the overall theme of how organic compounds function and interact. So, recognizing these structural features helps us appreciate their diversity and importance in chemistry and biology. Let's take a closer look at how carbohydrates are classified based on their behavior during hydrolysis. On this slide, we see three main categories: polysaccharides, oligosaccharides, and monosaccharides. Polysaccharides are large, complex carbohydrates made up of many sugar units linked together. Think of them as the long chains or networks, like starch or cellulose, which are important energy sources and structural components. Oligosaccharides are shorter chains, typically composed of a few sugar units, say 3 to 10. They often have specialized roles, such as cell recognition and signaling, and are found in foods like onions and garlic. Finally, monosaccharides are the simplest form, single sugar molecules like glucose or fructose. These are the fundamental units that make up more complex carbohydrates, and they are readily absorbed by our body to provide quick energy. The key idea here is that this classification hinges on how these carbohydrates break down during hydrolysis, meaning when they are chemically broken apart with water. Larger structures like polysaccharides break down into oligosaccharides, which in turn break down into monosaccharides. Understanding this breakdown process is crucial because it helps us grasp how our body digests and utilizes carbohydrates, fitting into the bigger picture of nutrition and metabolism. Let's talk about monosaccharides. These are carbohydrates that cannot be broken down further into simpler units like aldehydes or ketones. Essentially, they are the basic building blocks of more complex sugars. In nature, we know about around 20 different monosaccharides, which occur naturally in various forms. Some of the most common examples include glucose, which is our primary source of energy, fructose found in fruits, and ribose, an important component of nucleic acids. Understanding these simple sugars is crucial because they form the foundation of carbohydrate chemistry and are integral to many biological processes. As we explore the broader world of carbohydrates, recognizing these key monosaccharides helps us see how complex sugars are built and how they function in living organisms. Let's look at how some disaccharides break down. Maltose, for example, when it reacts with water, yields only 2 glucose molecules. Similarly, sucrose reacts with water to produce glucose and fructose. These are classic examples of disaccharides, which are a specific type of oligosaccharide. Now, oligosaccharides generally refer to carbohydrates that, upon hydrolysis, give us between 2 and 10 monosaccharide units. They can be classified further based on how many monosaccharides they produce: disaccharides, trisaccharides, tetrasaccharides, and so on. Most commonly, we encounter disaccharides, which consist of 2 monosaccharide units. The interesting part is that these units can be the same or different, depending on the specific disaccharide. Overall, understanding these smaller carbohydrate units is crucial because they're the building blocks for larger, more complex carbohydrates, which play significant roles in energy storage and structural functions within living organisms. Polysaccharides are interesting because they are not sweet in taste, which is why they are also called non-sugars. They tend to be amorphous, meaning they don't have a defined shape, and they are insoluble in water, making them quite different from simple sugars. When we look at a process like starch interacting with water, it breaks down into glucose units, with the general formula being N times glucose. Some common examples of polysaccharides include starch, cellulose, glycogen, and gums. These are all complex carbohydrates that, when hydrolyzed, yield a large number of monosaccharide units. This is a key concept because it highlights how they serve as energy storage or structural components in living organisms. In essence, polysaccharides are large, complex molecules that play vital roles in biological systems, and their properties, like being non-sweet and insoluble, distinguish them from simpler sugars. Understanding these points helps us see their importance in nutrition and biology, fitting into the larger theme of carbohydrate diversity and function. Let's take a look at how carbohydrates are classified based on their nature. On this slide, we have a table that divides them into 2 main categories: reducing sugars and non-reducing sugars. Reducing sugars are all those carbohydrates that can reduce failing solution and Tollens reagent. This means they have a free aldehydic or ketonic group that reacts during these tests. For example, glucose and fructose are classic reducing sugars because they contain these free groups. On the other hand, non-reducing sugars do not have this ability. They cannot reduce failing solution or Tollens reagent because they lack a free aldehyde or ketone group. Sucrose is a common example here. It's a disaccharide made from glucose and fructose, but its glycosidic linkage prevents it from acting as a reducing sugar. So the key takeaway is that the presence or absence of a free aldehydic or ketonic group is what determines whether a carbohydrate is a reducing or non-reducing sugar. This classification is fundamental because it helps us understand the chemical behavior of different sugars and their roles in biological systems. Now, let's talk about how monosaccharides are classified based on their functional groups and the number of carbon atoms they contain. If a monosaccharide has an aldehyde group, we call it an aldose. On the other hand, if it has a keto group, it's known as a ketose. These groups are key because they determine the chemical behavior and reactivity of the sugars. Understanding whether a sugar is an aldose or a ketose helps us predict how it will interact in biological systems. This classification is fundamental as we explore the diversity of sugars and their roles in life processes. So remember, it's all about that functional group and the number of carbons, which together define the type of monosaccharide we're dealing with. Today, let's talk about glucose and its key properties. First, glucose has a sweetened taste, which makes it easily recognizable and appealing in many food products. It's also soluble in water, meaning it dissolves readily, making it very useful in various biological and industrial processes. When you look at glucose in a laboratory setting, you'll see that it appears as a colorless crystalline solid, which is quite characteristic of many simple sugars. Understanding these basic properties helps us grasp how glucose functions both biologically and commercially. Now, moving on, we'll explore how glucose is prepared and its specific structural features, which are fundamental to understanding its role in energy metabolism and other biochemical pathways. Glucose occurs freely in nature as well as in combined forms. You can find it naturally in sweet fruits and honey, which makes it a common source of energy in our diet. Ripe grapes, for example, contain large amounts of glucose, giving them their sweet taste. When we talk about the preparation of glucose, it's important to understand these natural sources, as they form the basis for extracting or synthesizing glucose in the lab. This natural abundance also highlights the significance of glucose in biological processes and its role as a fundamental energy source. So, in summary, glucose is not only vital for our bodies, but also widely available in nature, which makes its preparation both interesting and essential for various applications. Glucose is an aldohexose, which means it has 6 carbons and an aldehyde group. It's also commonly known as dextrose, a term you might have seen in food labels. This molecule is fundamental because it's the monomer for many larger carbohydrates, such as starch and cellulose. These are essential for storing energy and providing structural support in plants. Interestingly, glucose is probably the most abundant organic compound on Earth, highlighting just how vital it is in biological systems. It belongs to the D series of sugars, which refers to the specific spatial configuration of its chiral centers. And it's a dextrorotatory compound, meaning it rotates plane polarized light to the right. Now, when we look at the structure of glucose, we're going to see how these features come together to define its properties and reactivity, which are key to understanding its role in metabolism and biochemistry. This foundational molecule links many processes in life sciences, tying into our overall theme of understanding biological molecules at the molecular level. Let's start with the first set of points. When glucose reacts with a mild oxidizing agent like bromine water, it gets oxidized to a 6-carbon carboxylic acid called gluconic acid. This reaction indicates that glucose has an aldehydic group, which is essential for this oxidation process. Moving on, if we heat glucose with hydrogen iodide, or HI, for a prolonged period, it eventually forms N-hexane. This tells us that all 6 carbon atoms in glucose are linked together in a straight chain, confirming its open-chain structure in certain conditions. Next, when glucose is oxidized with nitric acid, both glucose itself and gluconic acid produce a dicarboxylic acid known as saccharic acid. This is significant because it shows glucose contains a primary alcoholic hydroxyl group, which can be oxidized to form this dicarboxylic acid. Now, let's look at some reactions that confirm the presence of a carbonyl group in glucose. When glucose reacts with hydroxylamine, it forms an oxime, and when it reacts with hydrogen cyanide, it forms cyanohydrin. These reactions are classic indicators of a carbonyl group, specifically a C=O group, in the molecule. Finally, the molecular formula of glucose is C6H12O6. All these pieces of evidence, oxidation reactions, reactions with hydroxylamine and cyanide, help us understand the structure of glucose and confirm its key functional groups. Together, they paint a clear picture of glucose's molecular framework and its reactive nature. Glyceraldehyde exists in two enantiomeric forms, which are essentially mirror images of each other. This is a common feature in many biological molecules, and it plays a crucial role in how these molecules interact within living systems. Now, what's interesting about glyceraldehyde is that it contains just one asymmetric carbon atom. This single chiral center is what gives rise to the two different enantiomers, often labeled as D and L. Speaking of these notations, D and L are used to describe the orientation of the molecule, specifically based on the structure of the base glyceraldehyde. These notations help us distinguish between the different stereoisomers, which can have vastly different biological activities despite having the same molecular formula. Understanding these concepts is key as we explore how molecules like glyceraldehyde influence biological processes. Recognizing the importance of chirality and enantiomers helps us appreciate the complexity and specificity of biochemical interactions. Let's talk about the structure of glucose and some interesting features it has. So, this behavior couldn't be explained just by looking at the open chain structure of glucose. It was proposed that one of the OH groups might actually add to the CHO group, forming a cyclic hemiacetal structure. What's fascinating is that it turns out glucose forms a six-membered ring, where the OH group at carbon 5 is involved in creating this ring. This explains why the CHO group is no longer present in the same way, because it's now part of that ring system. This cyclic structure is also the reason why glucose exists in two different forms. We call these the alpha and beta forms. The alpha form has a melting point of about 419 Kelvin, and is obtained when you crystallize glucose from a concentrated solution at around 303 Kelvin. On the other hand, the beta form melts at about 423 Kelvin and forms when you crystallize from a hot, saturated aqueous solution at about 371 Kelvin. So, in summary, glucose's ability to form a six-membered ring explains a lot about its chemical behavior and physical forms, which is fundamental to understanding its role in biological systems and its chemical properties overall. Let's talk about mutarotation, which is really fascinating. It's the change in the optical rotation that we observe when the equilibrium between two anomers shifts. Essentially, this happens because the stereocenters in the sugar molecules are interconverting. When dealing with cyclic sugars, this process is especially noticeable because the alpha and beta anomeric forms are constantly converting back and forth. This interconversion causes the optical rotation to change, which is a key concept in understanding sugar behavior in solution. Recognizing mutarotation helps us see how sugars can exist in dynamic forms, contributing to their chemical properties and reactivity. So, mutarotation isn't just a curious phenomenon. It's a fundamental aspect of sugar chemistry that ties into the larger picture of stereochemistry and molecular behavior. Let's take a closer look at the two cyclic hemiacetal forms of glucose. These forms differ only in the configuration of the hydroxyl group at the C1 position, which is known as the anomeric carbon. The anomeric carbon is actually the aldehyde carbon before cyclization, and its configuration determines whether we have the alpha or beta form. These types of isomers are called anomers. Now, when we talk about the cyclic structure of glucose, especially the six-membered ring, we call it a pyranose structure. Think of it as being similar to purine, which is a cyclic organic compound with 1 oxygen atom and 5 carbon atoms in the ring. To represent this cyclic structure more accurately, we often use the Haworth structure, which provides a clearer depiction of the stereochemistry at each carbon in the ring. Understanding the concept of the anomeric carbon and the different anomers is important because they influence the reactivity and properties of glucose, and they tie into the overall theme of carbohydrate structure and function. This compound belongs to the D series, which means it's a naturally occurring stereoisomer, and it's levorotatory, so it rotates plane polarized light to the left. Fructose is quite interesting because it exists in 2 different cyclic forms, known as anomers. These forms are interconvertible, and the specific shape it takes can influence how it behaves biologically. When we look at the structure of fructose, or C6H12O6, we see that it's a 6-carbon sugar, but unlike glucose, its key feature is the ketone group, which classifies it as a ketohexose. This structure is fundamental to understanding its reactivity and role in metabolism. So overall, this slide highlights the stereochemistry, the cyclic nature with different anomers, and the chemical structure that underpins fructose's biological significance. So here we see an example where 2 monosaccharides are connected by a glycosidic linkage. Specifically, this linkage forms between the C1 carbon of an alpha-glucose molecule and the C4 carbon of another alpha-glucose. Now, let's talk about what a glycosidic linkage really is. It's essentially an oxygen atom that joins these 2 sugar units together. This linkage is formed through a chemical reaction where a water molecule is lost, a process called a dehydration reaction. When this happens, an oxygen atom acts as a bridge, connecting the 2 monosaccharides. This is important because such linkages are the foundation of disaccharides, which are basically 2 sugar units linked together. Understanding these linkages is key to grasping how complex carbohydrates are built and how they function in biological systems. So, in summary, a glycosidic linkage is this special oxygen bridge that forms between monosaccharides when a water molecule is removed, creating a disaccharide. This concept is fundamental to understanding carbohydrate structure and their biological roles. Let's now look at some important disaccharides and their properties. First, we have lactose, which is commonly known as milk sugar because it's found in milk. It's made up of beta-D-galactose and beta-D-glucose molecules. The linkage occurs between the C1 of galactose and the C4 of glucose. Since the anomeric carbon of one of the monosaccharides remains free, lactose is classified as a reducing sugar. Moving on to maltose, another disaccharide composed of 2 alpha-D-glucose units. In maltose, the C1 of one glucose links to the C4 of the other. The free aldehyde group at C1 of the second glucose makes it a reducing sugar as well. Now, sucrose is a bit different. It's a non-reducing sugar because the glycosidic linkage involves the C1 of alpha-glucose and C2 of beta-fructose, which involves their reducing groups. When sucrose undergoes hydrolysis, it produces equal amounts of D plus glucose and D fructose. An interesting point here is that sucrose is dextrorotatory, but after hydrolysis, the mixture becomes levorotatory. This is because fructose's levorotation is more dominant than glucose's dextrorotation, resulting in invert sugar, which has a negative rotation. Also, note that sucrose has no free anomeric carbon, because the linkage involves the anomeric carbons of both monosaccharides, making it a non-reducing sugar. Finally, the glycosidic linkage is a key concept here. It's the bond formed between 2 monosaccharides through an oxygen atom, resulting from the loss of a water molecule. This linkage is fundamental in forming disaccharides and other complex carbohydrates, connecting the monosaccharide units into larger structures. Polysaccharides are essentially large, complex carbohydrates made up of many monosaccharide units. These units are linked together by glycosidic bonds, which are special types of covalent bonds. Now, what's interesting is that polysaccharides are the most common carbohydrates you'll find in nature. They're everywhere around us, from the plants we eat to the structural components in various organisms. Most importantly, polysaccharides serve 2 main roles. One is as food storage. Think of starch in plants or glycogen in animals, providing a reserve of energy that the organism can draw on later. The other is as structural materials, like cellulose in plant cell walls or chitin in fungi and insects, that give support and shape to the organism. So, understanding polysaccharides helps us see how nature efficiently stores energy and builds structural frameworks, which ties into the broader theme of how biological systems manage resources and maintain integrity. Now, let's take a closer look at starch, which is a crucial energy source for us humans. Starch is made up of two main components, amylopectin and amylose. Both are polymers of alpha-glucose, but they differ in structure and how they are arranged. Amylopectin is highly branched, making it more easily digestible, while amylose is mostly linear. These differences affect how our bodies break down starch and absorb glucose. Starch is the most important dietary source of carbohydrates for us. You'll find it abundantly in cereals like rice and wheat, as well as in roots and tubers such as potatoes and yams, and even some vegetables. In plants, starch serves as the main storage polysaccharide, storing energy that the plant can draw upon when needed. So, understanding starch is key because it links plant biology to our nutrition and energy intake. Now, let's take a closer look at the two main components of starch. First, we have amylopectin. It's insoluble in water and makes up about 80 to 85% of starch, which is quite significant. Amylopectin is a branched chain polymer composed of alpha-D-glucose units. The key thing to remember here is how the chain is formed. It's connected mainly through C1 to C4 glycosidic linkages, but the branches occur via C1 to C6 linkages. This branching structure is what provides the unique properties of amylopectin, like its digestibility and how it interacts with water. On the other hand, we have amylose, which accounts for roughly 15 to 20% of starch. Unlike amylopectin, amylose is a long, unbranched chain of glucose units, typically between 200 and 1,000 units, linked through C1 to C4 glycosidic bonds. Because it's unbranched, amylose tends to be less soluble in water, but it plays a crucial role in the overall structure and digestibility of starch. Understanding these two components helps us see how starch functions both biologically and industrially. Amylopectin's branch structure makes it more water-soluble and easier for enzymes to break down, while amylose's linear chains influence the texture and slow down digestion, which is important in food science. So, both components work together to give starch its unique properties that are vital in our diets and various applications. Cellulose is a straight-chain polysaccharide, meaning it's made up of long, unbranched chains. It's composed entirely of beta-D-glucose units, which are linked together in a specific way. The linkage occurs between the C1 carbon of one glucose molecule and the C4 carbon of the next, forming a glycosidic bond. This particular linkage gives cellulose its rigidity and structural strength. Cellulose is found exclusively in plants and is actually the most abundant organic substance in the plant kingdom. If you think about the plant cell wall, cellulose is a major component. It's what provides the structural support and shape to plant cells. So, understanding cellulose helps us appreciate how plants maintain their form and how they grow. In the broader context of our discussion on carbohydrates, cellulose is a perfect example of how structure directly influences function, especially in the plant world. It's fascinating to see how nature uses such a simple building block to create something so strong and vital for plant life. Glycogen is primarily stored in the liver, muscles, and brain, serving as a vital energy reserve. When our body needs a quick source of glucose, enzymes come into play and break down glycogen into glucose molecules that can be easily used for energy. Interestingly, glycogen isn't unique to humans. It's also found in yeast and fungi, highlighting its importance across different organisms. Sometimes called animal starch, glycogen's structure is quite similar to amylopectin, a carbohydrate found in plants. However, glycogen is more highly branched, which allows for rapid breakdown and release of glucose when needed. In essence, the carbohydrates stored in our bodies as glycogen are crucial for maintaining energy levels, especially during fasting or intense activity. This storage form helps us keep our energy supply steady and ready to be mobilized whenever necessary. Carbohydrates are found throughout the biosystem, often in combination with proteins and lipids. They play a crucial role in many biological processes. For example, two important aldopentoses, D-ribose and 2-deoxy-D-ribose, are integral components of nucleic acids like DNA and RNA. We also use carbohydrates in our daily lives, building furniture from cellulose, which is the main component of wood, or making clothing from cotton fibers. These materials are vital raw materials for industries such as textiles, paper production, lacquers, and breweries. Carbohydrates serve as storage molecules too. Plants store energy as starch, while animals store glycogen. The cell walls of bacteria and plants are primarily made of cellulose, giving structural support and protection. Honey, a natural carbohydrate, has been used for centuries as an instant energy source, especially in traditional Ayurvedic medicine. Overall, carbohydrates are essential for life, both in plants and animals. They form a significant part of our diet, providing energy and supporting various biological functions. Their importance cannot be overstated, as they are fundamental to the structure and energy needs of living organisms. Proteins are the most abundant biomolecules in living systems. They are found all over the body and are essential to our structure and functions. You can find their main sources in everyday foods like milk, cheese, pulses, peanuts, fish, and meat. These molecules are fundamental because they make up most of our body tissues and are crucial for growth and maintenance. The word protein actually comes from the Greek word proteos, meaning primary or of prime importance, which highlights just how vital they are. All proteins are made up of polymers of alpha amino acids, which are their building blocks. This means that proteins are essentially long chains of amino acids linked together in specific sequences, giving them unique structures and functions. Understanding proteins helps us see how our bodies are built and how they operate at a molecular level. They are truly the foundation of life structure and function. Let's talk about glycine. As with most amino acids, it's commonly represented by a 3-letter symbol, but sometimes you'll also see a 1-letter code. Now, all alpha amino acids have these simple, often memorable names. These names usually give us a clue about their properties or where they come from. For example, glycine gets its name because it has a sweet taste. The word glycos in Greek means sweet. Interestingly, tyrosine was first isolated from cheese, and tyros means cheese in Greek. Amino acids, in general, contain two main functional groups: the amino group, which is NH2, and the carboxyl group, which is COOH. The position of the amino group relative to the carboxyl group determines how we classify the amino acid, whether it's alpha, beta, gamma, delta, and so on. When we hydrolyze proteins, only the alpha amino acids are obtained, which are the ones most relevant to our discussion. It's also worth noting that amino acids can have other functional groups attached, which adds to their diversity and functionality. Connecting this to our overall theme, understanding these basic structures and classifications helps us grasp how amino acids build proteins and influence biological functions. Let's take a closer look at the classification of amino acids. First, we have the basic amino acids, which include lysine, arginine, and histidine. These are called basic because they tend to have a high pH and can accept protons. Next, we have the acidic amino acids, such as aspartic acid and glutamic acid. Now, what's really interesting is how amino acids are classified as acidic, basic, or neutral. This depends on the relative number of amino groups and carboxyl groups in their molecules. If the molecule has an equal number of amino and carboxyl groups, it's considered neutral. If there are more amino groups than carboxyl groups, the amino acid is classified as basic because it tends to accept protons. Conversely, if there are more carboxyl groups than amino groups, the amino acid is acidic since it tends to donate protons. Understanding this classification helps us see how amino acids behave in different environments, which is crucial for understanding protein structure and function. It ties into the overall theme of how amino acids influence protein chemistry and interactions. Let's start by talking about what happens to amino acids in aqueous solution. The carboxyl group can lose a proton, while the amino group can accept a proton. This results in a special form called a Zwitter ion, which is neutral overall, but contains both positive and negative charges. This is a key concept because it shows how amino acids can behave as amphoteric molecules, meaning they can react with both acids and bases. Now, moving to their physical properties, amino acids are typically colorless, crystalline solids. They are water-soluble, which is important because it allows them to participate easily in biological processes. They have high melting points and tend to behave more like salts rather than simple amines or acids. This is because they contain both an acidic carboxyl group and a basic amino group within the same molecule, giving them unique chemical behavior. Next, let's differentiate between types of amino acids based on how our bodies manage them. The amino acids that our bodies can synthesize on their own are called non-essential amino acids. There are 11 of these. On the other hand, there are amino acids that our bodies cannot produce, so we need to obtain them through our diet. These are called essential amino acids, and there are 9 of them. Finally, it's important to understand how amino acids are classified. This classification helps us understand their roles and how they contribute to protein structure and function. Throughout this presentation, keep in mind that these fundamental properties and classifications are the building blocks for understanding the broader role of amino acids in biology and health. These amino acids exist in both D and L forms, but most naturally occurring amino acids are in the L configuration. When we write the L form, the NH2 group is typically placed on the left-hand side of the structure. It's important to note that, except for glycine, all naturally occurring alpha amino acids are optically active. This is because the alpha carbon atom in these amino acids is asymmetric, meaning it has 4 different groups attached to it, creating a chiral center. Glycine stands out because it has a hydrogen as its side chain, which makes its alpha carbon symmetric, so it's not optically active. This asymmetry is a key feature because it influences how amino acids behave in biological systems and how they interact with light, which is fundamental to understanding their roles in proteins. Let's look at some examples to clarify these concepts. For instance, insulin, which contains 51 amino acids, is classified as a protein. Now, polypeptides with fewer amino acids are often called proteins if they typically have a well-defined 3-dimensional structure characteristic of proteins. When we talk about smaller linked amino acids, like 4, 5, or 6, we give specific names: tetrapeptide, pentapeptide, or hexapeptide. Once the chain exceeds 10 amino acids, we generally refer to the product as a polypeptide. And if a polypeptide chain has more than 100 amino acids with a molecular mass over 10,000 units, it is classified as a protein. Now, to give you an example, when a third amino acid joins a dipeptide, the resulting molecule is called a tripeptide, which contains 3 amino acids connected by 2 peptide bonds. For instance, if the carboxyl group of glycine reacts with the amino group of alanine, we get glycoalanine, a dipeptide. It's important to remember that proteins are polymers of alpha amino acids, connected through peptide bonds, which are a type of amide bond. Chemically, a peptide linkage forms between the carboxyl group of one amino acid and the amino group of another. Understanding this structure is fundamental because it underpins how proteins are built and function in our bodies. Let's take a closer look at globular proteins. Common examples include insulin and albumins. These proteins have a structure that results when the chains of polypeptides coil around each other, forming a spherical shape. Because of this compact, rounded structure, globular proteins are usually soluble in water, making them important in various biological processes. On the other hand, we have fibrous proteins. Examples include keratin, which is present in hair, wool, and silk, and myosin, found in muscles. These proteins have their polypeptide chains running parallel and are held together by hydrogen bonds and disulfide bonds, creating a fiber-like structure. Due to this arrangement, fibrous proteins tend to be insoluble in water, giving them strength and durability. So, in summary, based on their molecular shape, proteins are classified into these two main types, globular and fibrous. Each type has distinct structural features and solubility properties that are crucial to their functions in the body. Some proteins are made up of 2 or more polypeptide chains, which we call subunits. The way these subunits are arranged in space relative to each other is known as the quaternary structure. This level of structure is important because it influences the protein's overall shape and function. Moving on to the secondary structure, this refers to the specific shapes a long polypeptide chain can adopt. The two main types are the alpha helix and the beta-pleated sheet. These structures form due to regular folding of the backbone, stabilized by hydrogen bonds between the C=O and NH groups of the peptide bonds. Next, the tertiary structure represents the overall folding of the entire polypeptide chain, essentially a further level of organization beyond the secondary structures. This gives rise to two major shapes, fibrous and globular proteins. The stability of these structures relies on various forces, including hydrogen bonds, disulfide linkages, van der Waals forces, and electrostatic interactions. Focusing on the beta structure, all the peptide chains are extended to their maximum length and laid side by side, held together by hydrogen bonds. This arrangement resembles the pleated folds of drapery, so we call it a beta-pleated sheet. In contrast, the alpha helix forms when the polypeptide twists into a right-handed screw. This helix is stabilized by hydrogen bonds between the NH group of one amino acid and the C=O group of an amino acid a few residues away, creating a coiled structure. It's also important to remember that proteins can have one or multiple polypeptide chains. Each chain has a specific sequence of amino acids, which we call the primary structure. Any change in this sequence can lead to a different protein, highlighting how crucial the primary structure is to the protein's identity and function. Finally, as we've seen, the primary, secondary, tertiary, and quaternary structures are all interconnected, each level building on the previous one and becoming more complex. Understanding the structure and shape of proteins helps us grasp how they perform their diverse functions in the body. And to wrap up, there's a key difference between fibrous and globular proteins. Fibrous proteins are usually long, thread-like, and provide support and structure, while globular proteins are more compact, rounded, and often serve as enzymes or transport molecules. Each type has a unique role based on its shape and structure. Let's look at some common examples of protein denaturation. For instance, when you boil egg white, it coagulates and turns solid. Similarly, milk curdles due to the formation of lactic acid caused by bacteria. These are everyday examples that illustrate how proteins change when they are heated or undergo chemical changes. During denaturation, the higher-level structures of proteins, specifically the second and third structures, are destroyed. However, the primary structure, which is the sequence of amino acids, remains intact. This is an important point because it shows that the protein's fundamental building blocks are still there, even though its shape and function are lost. When a protein in its native, functional form faces physical changes like a temperature increase or chemical changes, such as pH shifts, the hydrogen bonds that maintain its shape get disturbed. As a result, the globular forms of the protein unfold and the helices uncoil. This process leads to a loss of the protein's biological activity, meaning it can no longer perform its specific functions. This entire process is what we call denaturation of proteins. It's essentially a structural change that impacts the protein's activity without breaking the primary amino acid sequence. Now, it's important to remember that a native protein is one that exists in a specific three-dimensional structure within a biological system. This structure is crucial because it directly relates to its biological activity. When denatured, the protein loses this specific shape, and consequently, its function. Let's talk about enzymes, which are almost all globular proteins. These fascinating molecules are incredibly specific. They only catalyze particular reactions and work with specific substrates. You might notice that enzymes are usually named after the compound or class of compounds they act upon, making it easier to understand their function. Enzymes are biological molecules, mainly proteins, that dramatically speed up the chemical reactions happening inside our cells. Without them, many reactions would occur too slowly to sustain life. They play crucial roles in processes like digestion and metabolism, helping our bodies function smoothly. It's also important to remember that biocatalysts like enzymes operate under very mild conditions. Think normal body temperature and neutral pH, making them perfectly suited for the delicate environment inside living organisms. So, in essence, enzymes are vital biological catalysts, protein molecules that enable our bodies to carry out essential functions efficiently and effectively. Let's take a closer look at enzyme naming conventions. Note that the ending of an enzyme's name is typically -ase. Now, focusing on oxidortictases, these are special enzymes that catalyze oxidation reduction reactions. Essentially, they facilitate the transfer of electrons by oxidizing one substrate while simultaneously reducing another. This dual action is what makes them unique. For example, the enzyme itself is called an oxidortictase. Understanding these enzymes is crucial because they play a vital role in many metabolic pathways, ensuring that energy transfer and electron flow happen efficiently within the cell. This concept ties into our overall theme of enzyme function and how enzymes drive essential biochemical reactions. Think of the enzyme and substrate as having a lock and key arrangement. The substrate fits precisely into the enzyme's active site, forming a temporary complex known as the enzyme substrate, or ES complex. Once bound, the enzyme facilitates internal rearrangements within this complex, leading to the formation of the product. An important point here is that the enzyme isn't consumed in the process. It gets regenerated, ready to bind to another substrate molecule. This mechanism highlights how enzymes are highly specific and efficient catalysts, playing a crucial role in speeding up biochemical reactions essential for life. Vitamins are actually complex organic molecules that our bodies can't produce on their own. Because of that, we need to get them through our diet. Even though they are required in small amounts, they play a crucial role in supporting essential metabolic reactions. These reactions are vital for our body's normal growth and maintenance. So, in the context of overall health, ensuring we get enough vitamins is fundamental to keep our bodies functioning properly and to support our development over time. Let's discuss fat-soluble vitamins first. These include vitamins A, D, E, and K. They are soluble in fats and oils, which means they are absorbed along with dietary fats. Because of this, they are stored in the liver and adipose tissues, allowing the body to draw on these stores when needed. This storage capability also means they don't need to be consumed every day, but excess intake can lead to toxicity. Now, moving on to water-soluble vitamins. These are vitamins B1, B2, B6, B12, and C. They dissolve in water, which makes them easier to absorb, but also means they are excreted in urine fairly quickly. Therefore, they need to be supplied regularly through our diet, except for vitamin B12, which has a unique storage capacity in the liver. Let's look at some common sources and deficiency diseases. Vitamin C, found in citrus fruits, amla, and green leafy vegetables, is crucial in preventing scurvy, which causes bleeding gums. Vitamin D, obtained from sunlight exposure, fish, and egg yolk, helps prevent rickets in children and osteomalacia in adults, both conditions related to soft or deformed bones. Vitamin E, present in milk, ghee, and vegetable oils like wheat germ, sunflower, and cottonseed oil, is vital for maintaining the integrity of red blood cells and muscle health. A deficiency can lead to increased fragility of RBCs and muscular weakness. Vitamin H, also known as biotin, found in milk, yeast, liver, and kidney, helps prevent hair loss and dermatitis when deficient. Vitamin K, sourced from green leafy vegetables, fish, meat, and cereals, is essential for proper blood clotting. A deficiency here can lead to increased bleeding tendencies. Moving to specific vitamins and their sources, vitamin A is abundant in fish liver oil, milk, butter, egg yolk, and green and yellow vegetables. Its deficiency causes night blindness and a condition called xerophthalmia, where the cornea hardens. Vitamin B1, or thiamine, found in yeast, milk, green vegetables, cereals, fruits, and egg yolk, prevents beriberi, which leads to loss of appetite and slowed growth. Vitamin B2, present in egg yolk, liver, milk, and leafy greens, is important for skin and digestive health, and deficiency causes cracked lips, sore tongue, and digestive issues. Vitamin B6, found in milk, egg yolk, cereals, yeast, and legumes, is crucial for nervous system health. Its deficiency can cause nervous disturbances and convulsions. Lastly, vitamin B12, present in meat, fish, kidney, and eggs, is essential for red blood cell production, and deficiency can lead to pernicious anemia, characterized by a lack of hemoglobin in red blood cells. Understanding these vitamins, their sources, and deficiency symptoms helps us appreciate the importance of a balanced diet for maintaining good health. Nucleic acids are the polymers of nucleotides that are found in the nucleus of all living cells. They play a crucial role in transmitting hereditary characteristics, which means they carry our genetic information from one generation to the next. Additionally, nucleic acids are essential for the biosynthesis of proteins, the building blocks of life. Now, when we talk about the structure of nucleic acids, we're delving into how these molecules are built. Understanding their structure helps us grasp how they perform their functions. Nucleic acids are made up of long chains of nucleotides, each consisting of a sugar, a phosphate group, and a nitrogenous base. These building blocks are linked together in a specific way, forming a double helix in the case of DNA, for example. This structural organization is key to their ability to store and transfer genetic information accurately. It also connects to the broader theme of how molecular structures underpin biological functions. Recognizing the structure of nucleic acids gives us insight into the fundamental processes of heredity and protein synthesis that sustain life. Today, I want to talk about nitrogenous bases, which are essential components of our genetic material. These are nitrogen-containing biological compounds that play a crucial role in forming nucleosides. Essentially, nitrogenous bases are the building blocks that connect to sugars to create nucleosides, which then form the backbone of DNA and RNA. Understanding these bases is fundamental because they determine the genetic code and influence how genetic information is stored and transmitted. They include molecules like adenine, thymine, cytosine, guanine, and uracil, each with unique structures and functions. By grasping the structure and role of nitrogenous bases, we can better appreciate the molecular basis of genetics and how our cells encode information. These bases are truly the key to understanding the complexity of life at a molecular level. A unit formed by the attachment of a base to the one position of a sugar is known as a nucleoside. So essentially, when we take a sugar molecule, like ribose or deoxyribose, and attach a nitrogenous base to its one carbon, we create a nucleoside. This is a fundamental building block in nucleic acids, which are DNA and RNA. The key point here is that the nucleoside itself doesn't include the phosphate group. It's just the sugar and the base. Understanding this helps us see how nucleotides are formed, since nucleotides are basically nucleosides with one or more phosphate groups attached. So, in the context of the overall structure of genetic material, nucleosides are crucial because they form the backbone of the more complex molecules like DNA and RNA. Let's talk about RNA pairing first. In RNA, adenine or AU pairs specifically with uracil. Unlike DNA, where thymine pairs with adenine, RNA uses uracil instead. Now in DNA, adenine pairs with thymine through 2 hydrogen bonds, which is represented as A equals T, and guanine pairs with cytosine through 3 hydrogen bonds, or G equals C. These hydrogen bonds are crucial because they stabilize the DNA double helix structure. An important concept here is the balance between purine and pyrimidine bases. The amount of purines, which are adenine and guanine, always equals the amount of pyrimidines, like thymine and cytosine. This pairing maintains the uniform width of the DNA double helix. To put it simply, the purine base on one strand always pairs with a pyrimidine base on the opposite strand. Now, shifting gears a little, when a nucleoside links to phosphoric acid at the 5' position of the sugar moiety, we get what's called a nucleotide. This linkage is fundamental because nucleotides are the building blocks of nucleic acids, like DNA and RNA. So, understanding this linkage helps us grasp how genetic information is stored and transferred at the molecular level.